All right, today we're cracking open one of the most important ideas in all of immunology, antigens. And to do it, we're going to use a simple, powerful analogy. Think of this as a top secret intelligence briefing where the human body is a sovereign nation and it's under constant threat. So let's set the scene. Picture your body as this sprawling, bustling country. Every single one of your cells is a citizen, just doing its job. And your immune system, well, that's your national security force. Highly trained, always on alert, protecting the borders and keeping the peace. Here's our game plan for this briefing. We'll start by exploring our country, the body. Then we'll get into identifying intruders and how we inspect them. From there, it's all about defense protocols, a serious code red threat, and finally, how we can boost our national security. Okay, let's dive in. Our country, the body, is always dealing with the outside world. And just like any nation, it has to have a way to know the difference between its own citizens and any foreigners that show up. This right here is the primary job of our security forces, the immune system. The single most important piece of intel we're looking at today is the antigen. So what is it? Think of it as an ID card, or a uniform, or even a flag. It's some kind of identification marker that everything, and I mean everything, carries around. It's the very first thing our security patrols check to figure out, are you a friend or are you a foe? Okay, so we know what an antigen is. Now we gotta figure out how to spot the real intruders. And here's something that's absolutely crucial to get. Just because something is carrying a foreign ID doesn't automatically make it a threat. Our security forces are smarter than that. They have to actually assess the level of danger. And that brings us to a really key distinction: immunogenicity versus antigenicity. See, a substance can be antigenic, which just means our security patrols recognize its ID. They can see it, but that doesn't mean it's immunogenic. To be immunogenic, it has to be seen as a real threat, one that provokes a full-blown response. Alarms blaring, troops mobilizing, the whole 9 yards. A perfect example of this is something called a hapten. A hapten is like a lone, petty thief. Yeah, it's a foreign entity, so it's antigenic. Our guards can see its ID. But on its own, it's just too small and insignificant to trigger a big security response. It's not immunogenic. This is actually the idea behind some drug allergies, where a tiny drug molecule is ignored until it finds a getaway driver. So how does this lone thief become a major threat? Well, it teams up with a bigger gang, a much larger molecule that we call a carrier. The second that hapten latches onto a carrier, the whole package suddenly looks big, suspicious, and dangerous enough to be immunogenic. And that's when you get that full-scale immune response. Now, these threats can pop up from a few different places. You've got your exogenous antigens. Those are the foreigners trying to cross the border, stuff we breathe in or eat. Then you've got endogenous antigens, which are like homegrown criminals, our own cells that have been corrupted by something like a virus. And then you've got autoantigens. This is a tragic case of friendly fire, where our security forces mistakenly tag loyal citizens as the enemy. And that's what leads to the chaos of autoimmune diseases. Okay, so we know where these threats are coming from. Now, let's zoom in and take a much closer look. When our security forces spot an intruder's uniform, they don't just glance at the whole thing. Nope. They are trained to focus on one very specific, tiny detail. And that specific detail? It's called the epitope. The epitope is that small, unique part of the antigen that our immune cells actually lock onto. So it's not the entire uniform they care about, but the specific badge sewn onto the sleeve. And get this, one antigen can have lots of different epitopes, just like a soldier's uniform can have multiple different badges and patches. Okay, now for everybody studying for an exam, listen up. This is a huge one, a line you absolutely have to burn into your memory. The epitope is the smallest unit of antigenicity. It is the most fundamental piece of ID our immune system ever looks at. So if the epitope is the badge, well, you need a scanner that's designed to read that specific badge, right? That scanner is the paratope. It's the part of our antibody or T-cell receptor that is perfectly shaped to lock on to the epitope. It's this incredibly precise lock-and-key fit that makes our immune system so darn good at its job. So let's move on to our defense protocols. You see, our national security system is not a one-trick pony. It doesn't have a one-size-fits-all solution. Instead, it customizes its strategy based on the exact kind of threat it's up against. We can basically group threats into two main profiles. First, you have T-dependent antigens. Think of these as an organized, invading army. They're complex, usually proteins, and they demand a full, coordinated military response. This response is powerful, and, this is key, it builds a long-term defense memory. On the other hand, you have T-independent antigens. These are more like scattered rioters. They're simpler, and our forces can handle them with quick police action. But, and this is a big but, that kind of response doesn't build any lasting memory for the next time. So far, we've talked about a pretty orderly system of checks and balances. But what happens when an intruder shows up that doesn't play by any of the rules? That, my friends, is a code red scenario. And that's when we have to talk about superantigens. Just think about it for a second. What if an intruder had a master key? What if it could just skip all the specific badge and scanner protocols and just hotwire the entire national alarm system, setting off every single siren in every city all at the same time? Well, that is exactly what a superantigen does. It's a type of antigen that doesn't need to be properly processed and presented. It just bypasses all the normal safety checks and activates a massive number of our T-cell security forces in a completely random, non-specific way. Here's how it pulls it off. A normal antigen needs that perfect, specific fit. But a superantigen acts like a universal bridge, forcing our security cells to link up when they absolutely shouldn't. This can trigger up to 20% of our T-cells all at once, and that leads to a massive, out-of-control release of communication signals called cytokines. This cytokine storm is the biological equivalent of mass panic and friendly fire, causing unbelievable chaos and collateral damage to our own country. And this isn't just some abstract threat. This chaos is the real-world reason behind some very serious conditions. The toxins that cause toxic shock syndrome and scalded skin syndrome, yep, those are superantigens. Same goes for the triggers behind conditions like Kawasaki syndrome. Okay, after talking about a threat that massive, let's switch gears and end on a more positive note. How can we actually boost our country's national security? How can we make our defense forces even better at their job? Well, one way we do it is with adjuvants. An adjuvant is a substance that basically makes an antigen more immunogenic. Think of adjuvants as intelligence amplifiers. They don't fight the battle themselves, but they basically grab our security forces by the shoulders and shout, "Hey, pay attention to this one. This one's important." And you can see the difference they make right here. On the left, you've got a standard response to a vaccine. It's good, it works. But on the right, you add an adjuvant, and the response gets amplified. More security forces show up, intel gets processed faster, and the result is a much stronger, more robust, and longer-lasting defense. It's a cornerstone of how many modern vaccines work so well. So what's the big takeaway from our briefing today? It's this. Antigens aren't automatically the bad guys. They are simply identity markers. They are the critical language that our body reads to make one of its most vital decisions: who gets a welcome pass and who needs to be eliminated. Understanding that language is the key to understanding immunology.